Welcome back guys, welcome back in another video tutorial from microbiology with Jagnik. So from now on, with the help of couple of videos, we will going to study in very much details about the Kelvin cycle. So let's get started with the first video of this journey. So first in introduction, first we have to learn that photosynthesis is take place in chloroplast, and it is very common to us. We are learning it from very basic foundation level of our education, right? So photosynthesis take place in chloroplast. Now this photosynthesis are of two types. One is light reaction, another one is the reaction which is not directly energized by light, or the dark reaction. So in light reaction, the energy conserved as a high energy phosphoanhydride bond of ATP or reducing power of NADPH. So all the protein and pigments responsible for light reaction located in the thylakoid or granadisk membrane. Okay, so here we will going to talk about the Kelvin cycle, which is the photosynthetic dark reaction. Now the first question which should come in our mind, that is, why Kelvin cycle? So Kelvin cycle is used to fix the CO2 and to reduce it to form hexose sugar or carbohydrate. Okay, now next question that should come in our mind, that is, what will be the driving source, or what will be the energy that will drive the reaction? So remember, the energy will come from the cleavage of the phosphoanhydride bond of ATP that we have just talked about, as well as the reducing power of NADPH. Okay, so all the enzyme and intermediates responsible for the Kelvin cycle are located in the chloroplast stroma, which looks like matrix of the mitochondria. Now the Kelvin cycle consists of mainly three stages. In the first stage, we will going to fix the CO2 with the help of ribulose bisphosphate, producing 3-phosphoglycerate, and it will be 2 molecule of 3-phosphoglycerate. Then reduction of this 3-phosphoglycerate to form hexose, and regeneration of the ribulose bisphosphate so that more CO2 can be fixed. Okay, so that is the idea. So now the main enzyme which going to play a very important role in the first part of the Kelvin cycle, that is the fixation of the CO2, that is ribulose bisphosphate carboxylase. Okay, and the reaction is ribulose 1,5 bisphosphate plus CO2 is equal to 2 molecule of 3-phosphoglycerate. But it is not the only reaction that ribulose bisphosphate carboxylase facilitates. Along with that, it also facilitates a oxygenase reaction, and that's why ribulose bisphosphate carboxylase is also known as ribulose bisphosphate carboxylase oxygenase, or RUBISCO, the most abundant enzyme on earth. Okay, it is very important for our MCQ purpose. Okay, so that is the idea. Now let's talk about the reaction mechanism of ribulose 1,5 bisphosphate plus CO2 is equal to 2 molecule of 3-phosphoglycerate. So here it is the reaction mechanism that I have written for you, and I will going to explain it with the help of a diagram. Before that, in this reaction mechanism, remember there is a intermediate named beta keto acid intermediate, and there are several other type of molecules present that look like this beta keto acid intermediate, and thereby those other molecules that looks like this beta keto acid intermediate act as a transition state analog. Okay, and that's why those other molecules have the ability to inhibit RUBP carboxylase activity. So we should learn and remember about the example of those type of molecules. So those type of molecules are 2-carboxy-arabinitol 1,5 bisphosphate and carboxy-arabinitol 1-phosphate. Okay, so now let's talk about the reaction mechanism. So this is ribulose 1,5 bisphosphate. First step, if you have watched the what I have written in the previous slide, so the first step was the extraction of H plus or proton from the C3. Okay, so as a result, a double bond will be introduced between the carbon number 2 and carbon number 3. Then nucleophilic attack on this carbon dioxide, giving us beta keto intermediate. Then water will be added. or it will be a hydrolysis reaction giving us 2 molecules of 3-phosphoglycerate. So very easy. So that's the idea. So now we will going to talk about the RUBP carboxylase in very much details. So remember, RUBP carboxylase usually consists of 2 subunits. Okay. In case of plants, so one subunit is large subunit which is present 8 in number, another one is 8 small subunit. And remember, the large subunits especially take place in the catalysis reaction. Okay. Now there are some bacteria that only have the large subunit, with the smallest subunit being a homodimer. Okay. Although it is very clear to us that the large subunit involved in catalysis, but it is not clear until now what is the role of these small subunits. But still there are some evidence that small subunit involved in regulation of the catalysis. Okay, they interact with the large subunit and facilitates regulation. Okay. Now we will going to talk about the large subunit in a little bit details, that the large subunit is actually a dimer that arranged in anti-parallel orientation. Okay, meaning the N terminal of one monomer will be adjacent to the C terminal of the other. Okay, and the active site will be at the interface between the monomers within the dimers. Okay, and the substrate binding site will be at the mouth of the alpha beta barrel domain of the large subunit. The very important thing that we have to remember, the previous 2 point is not too much important, but the very important thing is the active site of the ribulose bisphosphate carboxylase is surrounded by charged amino acids. Okay, this is very important. Why? You can remember, you can understand with the help of this slide. The slide tells us that when RUBP carboxylase in active condition, it should have a carbamate that binds an essential Mg2 plus at the active site. This Mg2 plus obviously will going to act as a cofactor. Now the question is how this carbamate is formed, which is very important for RUBP carboxylase to be in its active form. So carbamate is formed by the reaction of HCO3 minus with the epsilon amino group of the lysine amino acid, which is present in the active site of the enzyme in the presence of Mg2 plus. Remember, the HCO3 minus that reacting to form the carbamate and the CO2 that act as a substrate is distinct. Okay, and Mg2 plus act as a bridging substance between the oxygen of HCO3 minus and the substrate carbon dioxide. Okay, so now binding of either the main substrate, which is RUBP, or transition state analogues that we have just talked about at the active site of RUBP carboxylase causes a conformational changes of the enzyme. Okay, from its open state to closed state. Remember, the RUBP carboxylase enzyme can be in two conformation. Okay, when RUBP carboxylase is free of substrate, it will be in open state, and when RUBP carboxylase is having substrate in its active site, it will be in closed state. It will be in closed state with the substrate to prevent the access of solvent water to active site. Okay, but remember, the closed state is useful and very beneficial when this carbamate is present. Otherwise, it may be not a good news for that RUBP carboxylase and for the catalysis reaction. Why? Because remember that RUBP carboxylase can be spontaneously inactivated by decarbamylation reaction, that is the removal of carbamate. Okay, what happens when carbamate is not present? RUBP carboxylase will going to tightly bound this RUBP, that is the main substrate. Not only RUBP, it may also bind other sugar phosphate tightly in a dead-in complex formation at its closed state. Okay, and as a result, the entire reaction becomes inactive and there is no catalysis reaction occurring. Now what to do? So for reforming the carbamate, it is essential that enzyme should be back to into its open state conformation. Okay, and for coming back to the open state conformation, enzyme will going to play a very important role is the RUBP carboxylase activase, okay, which is an ATP hydrolysing enzyme. Okay, so now what this RUBP carboxylase activase will do? RUBP carboxylase activase will going to allow the enzyme to go back to its open state, so that all the RUBP carboxylase and sugar phosphates that was tightly bound with the RUBP carboxylase active site can be removed and the carbamate can be reformed, right? Along with that, remember that during the light reaction there is a production of ATP, and it was observed that there is a ATP dependence on Ribus activation. Okay, that also tell us that there should be another type of mechanism for Ribus activation, that is light dependence activation of the enzyme, but we will not going to talk about this. We will going to just remember about the ATPase activation, right, or ATPase mediated activation, right? So this ATPase is a AAA family of ATPases, and many of the member of this family have chaperone like roles. Now this ATPase is large multimeric protein subunit complexes that surround the Ribus during its job, that is the conformational changes into the open state of the enzyme. Okay, so that is the our. So here I have also mentioned the reaction that I am telling about, how carbamate is formed by reaction with the amino group present at the epsilon position of the lysine with the HCO3 minus. So that is how the carbamate is formed, mainly that is structure. Okay, so that's the idea of our first video. Today we have just learned about the how RUBP carboxylase works, what is the arrangement of RUBP carboxylase, and we have talked about the basic of Kelvin cycle. So in the next part of our video, we will going to talk about photorespiration, where we will going to learn that O2 may also compete with CO2 for binding with Ribus. That's why we are calling that Ribus as RUBP carboxylase oxygenase. That means it not only going to fix CO2, but it also have the ability to fix O2, right? That will be our next part of our video. Not only that, we will going to talk about some type of plants known as C4 plant. They are following another type of pathway for fixing carbon dioxide to minimize the photorespiration, right? And that's how we will going to learn the entire Kelvin cycle with the help of a couple of videos that will to come. Okay, so I hope this video will be helpful for you for understanding the basics of Kelvin cycle. So thank you for listening to this class.